So I'm really happy to share some of the projects our subcommittee has been working on and some of the progress we've made towards trying to solve the problem of drug-related thrombosis. So if we could have the next slide. Our committee is co-chaired by Eric Schultz, who's a pathologist and scientist at Eli Lilly. And my lab is located at Cornell University's College of Veterinary Medicine. And we really specialize on coagulation testing and research on coagulation disorders in animals. And you can have the next slide. We have a group of colleagues on our team who really, as we've heard before, represent industry, academia, and government laboratories. And it's really very helpful to have this varied expertise. Our team members participate by helping design studies, running assays, and of course, putting together the data and publishing. So if I can have the next slide. So the problem we're trying to solve now is the fact that current preclinical test practices are really not sensitive enough to accurately predict thrombotic risk of new drugs and new compounds. And what we'd like to do is develop a set of screening tests that are translational across species and that could be incorporated very early on in the toxicity test scheme. So if I can have the next slide. So of course, this is not a trivial problem because thrombosis is not one thing. Thrombosis accompanies many different disease syndromes, diverse anatomic sites and clinical picture depending on the site and the cause of thrombosis. And then if we look at the other side of the coin, well, what are the drugs and biologics that have been associated with thrombosis? It spans many different classes and many different drug indications. So in fact, you're treating very diverse patient populations. And unfortunately, as we've recently learned, thrombotic complications can develop in healthy people. And we saw that with the COVID-19 vaccine. So if we go on to the next slide. So our group started addressing the problem by performing a gap analysis. So there was a survey, and what we found is that thrombotic risk at present is really defined by the presence of gross thrombi that can be visualized at necropsy or on histologic sections. And pretty much by definition, you've already gone over the threshold of a procoagulant stimulus to a severe thrombotic insult, if that's your metric of thrombotic risk. The testing that is performed, though, is really designed to pick up hypocoagulability or a bleeding tendency. And there aren't comparable biomarkers that have been well validated in animals to detect a hypercoagulable or prothrombotic state. So we're kind of starting from square one. And if we go on to the next slide. So we started with a series of animal model studies, modeling different aspects and types of hypercoagulability. We chose the rat as our model species because they're often used early stage toxicity studies, and also rats are big enough so that we can get a lot of sample and do a lot of assays from a single individual. Our first project looked at a model of bacterial sepsis-induced thrombosis. And we, when an animal or a person has overwhelming sepsis, there's a combination of endothelial cell activation, white cell activation, and crosstalk between these different cell types with the hemostatic system. So you end up with an activated or procoagulant state. The next slide summarizes the results of this project. So what we did was try to dial down our endotoxin infusion and take serial samples over time so that we could capture changes in a wide variety of hemostatic proteins. And what we could piece together by looking at this time course study is that we saw early increases in procoagulant proteins, and what I thought was really interesting, very striking increase in procoagulant microparticles that are released from activated platelets and leukocytes. We also performed microRNA profiling, and that gave us a clue to some of the regulatory pathways that over time dampened down the clotting cascade and sort of brought hemostasis back into balance. So that was our first study. And starting with the next slide, our group wanted to look at the influence of pre-existing conditions in the patient population. And of course, when you think about it, toxicity studies are almost invariably performed in healthy young animals, whereas the patient population for many drugs is not a healthy young person. So we wanted to dial up the sensitivity of the test animal by examining an animal with pre-existing conditions. So in the next slide, we chose the Zucker diabetic fatty rat. These rats have a mutation in the leptin receptor that renders them susceptible to developing diabetes. And we divided the rats into two groups, one fed low fat, one fed high fat. And we monitored them over time to look for, predicted or expected metabolic derangements, but also, again, wide panel of hemostatic proteins and other biomarkers of coagulation balance. So the next slide shows our results. And we did see the expected overt manifestation of diabetes in the high fat group. So these rats were the same weight, same genetic background, but high fat induced a diabetic state. The other thing that was interesting is that we saw upregulation of some hemostatic proteins that also participate in inflammation. So fibrinogen, von Willebrand factor, and the anti-fibrinolytic protein PI1 were all significantly increased in these rats. And the other thing we did was perform functional testing, looking at thrombin generating capacity. So the next slide demonstrates the central role of thrombin in coagulation. And this, thrombin is very conserved. So these functions of thrombin in terms of transforming soluble fibrinogen into the fibrin clot. Thrombin is also a potent platelet activating agent. And thrombin participates in many feedback loops so that it actually encourages or high thrombin can spiral out of control where you generate more thrombin through activation of coagulation proenzymes. So what we found was that in addition to high circulating hemostatic proteins, in fact, the ZDF rats had an increased thrombin generating capacity. And again, this is recognized as a risk factor for venous thrombosis in people. So in fact, we had identified a very susceptible animal model that to study thrombotic risk. Next slide shows our study that we're working on now, where we really wanted to, and the group really wanted to refine the stimulus. So on the left side of the slide, we, in pink, we see the procoagulant enzymatic reactions that lead to the formation of thrombin and ultimately the fibrin clot. On the right-hand side, we see in blue the fibrinolytic pathway enzymes, and this system degrades the fibrin clot and ultimately reestablishes vascular patency. So in our study, what we want to do is directly activate coagulation by infusing tissue factor, which is the primary stimulator of this procoagulant response. And we'll also include an anti-fibrinolytic drug, tranexamic acid, so that we specifically block plasma-mediated fibrinolysis. So the tricky part is really fine-tuning dosage. And the next slide shows our pre-pilot, where we don't want to induce overt thrombosis, so we can't use the presence of thrombus as an endpoint. but we could use a very sensitive biomarker of thrombin excess, TAT complex, or thrombin antithrombin complex. And the panel on the right shows dose escalation studies where we use increasing doses of tissue factor from group 1 to group 4, and we saw a concomitant increase in TAT complex concentration in these treated rats. So it really looks like we have a nice system where we can dial up or dial down the thrombotic stimulus as we want to follow this procoagulant imbalance. So the next slide, kind of summarizes where do we want to take this. Well, what we'd like to do is develop a refined set of very sensitive screening tests that we could apply to early phase drug testing that would be translational across species, where we could detect a thrombotic signal, again, very early on in the drug development process. But we'd also like to have an expanded panel of biomarkers so that if we do detect a thrombotic signal, we can apply that panel and gain some mechanistic understanding. And potentially this could be useful not just for evaluating drugs, but also disease modeling. And if we make it translational, then we can really have a sort of very useful set of tools to evaluate thrombotic syndromes in any species and for any disease. So there's lots of opportunities for cross collaborations here in vascular biology and certainly immunology. And again, obviously hematology and computational biology. So my last slide is, again, call for collaborators and participants. So if you're interested or your group is interested in any aspect of hemostasis, come on and join us. We've been working with in vivo models, but there are some blood-on-chip technologies that I think that we could really incorporate. And we'd like to look at hemostasis under flow and varying shear stress. So again, please contact Jen or Alyssa and join us.
Dr. Brooks, listen, thank you very much for a great overview of this project, and also thank you for kind of getting us back on time because we're pretty far behind. I guess a quick question for you is how do you see, once you develop these assays, how would you trigger these tests in a drug development candidate? I mean, you wouldn't want to do them on every candidate. So do you see that as just kind of being driven by the target?
Yeah, I think you could take it any number of ways, and if we make it or refine it simple enough, at some point the drug has to go in the animal, and routinely tests like the APTT and PT and platelet count are incorporated. So if we had just a few potentially fast and cheap assays, they could be incorporated early on. But obviously, depending on the class of compound and your patient population, you might incorporate these tests earlier or later.
Thanks very much. I'm just looking to see if there's any, oh, here we go. This one is from Martha, and she's saying, do we understand why type 1 diabetes patients are different than type 2 diabetes patients? There is a mixed population for type 2 that are type 1s, but are type 2 characteristics that are not typical. Any commentary on that?
Yeah, I mean, I'm an internist, but don't deal with diabetes anymore. But for sure, the metabolic derangements in terms of lipid metabolism and lipid derangements are different in type 2 diabetes, where it's related to obesity and adiposity and that kind of thing, metabolic syndrome, versus the autoimmune problems. So I think, and also the age of onset and so on would be different in these patient populations.
Great. And I think maybe our last question will be from Dave Eaton. Is there any evidence that chronic inflammatory responses or pro-inflammatory cytokines contribute to coagulation problems?
Yeah, absolutely. So evolutionarily, it's really very interesting, but the complement pathway and the coagulation pathway are related, and there's a lot of crosstalk. So between innate immunity and hemostatic pathways for sure. And I think the COVID-19 syndrome of thrombosis has really made people look more closely at this crosstalk.